Mr. Majewski's Anatomy 32 class lecture, Chapter 16, Nervous Tissue. So, the nervous system is divided up between the central nervous system, which is the brain and spinal cord, and the peripheral nervous system, which are all the nerves that come off of the brain and spinal cord and penetrate our body. If you look at the nervous system in terms of function, you see that there is the sensory function, which is receptors that detect internal and external stimuli. These are called sensory or afferent neurons. For instance, they detect if your stomach is stretched, thereby you are full, or if somebody touched you on the arm. Then there's the integrative function. This is basically processing the information, storing it, making decisions to respond. Probably more of what we would think of as the actual thinking occurring. And these are called interneurons. And then there's the motor function, which is the response, such as to have a gland secrete hormones or to have a muscle contract. And these are called motor or efferent neurons. So, the peripheral nervous system is subdivided into a few categories. One is the somatic nervous system. This is the part of the nervous system that allows us to have voluntary control over things, usually skeletal muscles. And this has the somatic sensory pathways that take information from the outside world and bring it to the brain, and then the somatic motor pathways, which then send out a signal from the central nervous system to cause a response, such as to move your arm. Then there's the autonomic nervous system. This controls things that are involuntary, things that we don't really think about. So again, there are still autonomic sensory neurons functioning, sending signals to the central nervous system. For instance, if your stomach is stretched. And then you have the autonomic motor neurons then send the signal out from the central nervous system in order to do something, such as to, I guess in this case, make us not feel hungry, or to warn us when we're too full. So the autonomic nervous system can be broken up into two divisions. There's the sympathetic division. This is usually things that are stimulative, such as the heart rate speeding up. They often call it the fight or flight response, although we can do it separately. You don't necessarily have to have everything in the body speeding up. And then there's the parasympathetic division. This tends to be more of a relaxive thing. It slows down the heart rate and that sort of thing, often called the rest and digest responses. And then there's the third big nervous system, the enteric nervous system. This is also involuntary, and it's basically the nervous system of the gut. So our digestive tract can basically act independently of the central nervous system, although obviously normally signals are being sent to and from the central nervous system. So that means there are 4 separate neural networks in our body, and we'll be learning about the first 2 in greater detail, or I guess actually the first 3 in greater detail. So here's just another way to look at it. So you get the peripheral nervous system with its sensory components. That includes somatic, autonomic, and enteric sensory receptors and so forth. Then you have the central nervous system, where the signals of the senses are sent to. And then you have the motor components, the signals being sent from the central nervous system to then lead to responses such as skeletal muscle move and so forth. So this is a neuron. It has a body with a really, really long tail that's called an axon. It possesses the ability to be electrically excitable. So basically it can send a nerve impulse along, or action potential along its cell membrane to then lead to responses in other cells. So let's see. First of all, there's the cell body. The cell body has the normal organelles you'd expect, nucleus, mitochondria, so forth. But it also has a few unique structures, or uniquely named structures. There's the Nissl bodies. These are clusters of rough endoplasmic reticulum that are having such high levels of protein synthesis that they actually look distinct in the cell body. Then there's neurofibrils and microtubules. These are structures that are involved in the structure of the cell, as well as moving materials along the axon at a relatively rapid rate. Those would be the neurotransmitters. There's the lipofuscin, which is small yellowish-brown granules seen in the cell body. And then somatic spines. These are little bumps on the plasma membrane that increase the overall surface area of the cell body and of the dendrites. Which these are dendrites. They are protrusions off of the cell body that are usually relatively short. They receive information. They receive a signal. And they have many dendritic spines, which are pretty much the equivalent of the somatic spines. Then there's a long axon, usually only one, and this is what carries the signal away to the next cell or cells. It has a number of features. The axon hillock, where it's going from the cell body to the axon proper. The initial segment, which is just the first segment that's part of the axon hillock. You have the exoplasm, that's the cytoplasm specifically of the axon. Axolemma, which is the plasma membrane of the axon. Sometimes you have axon collaterals, those are side branches that can then go on and signal other cells. And also the axon terminal arborizations, which basically means that at the end of the axon it often spreads out into lots of little fibers. And here is some different ways that a neuron can appear. We've got the multipolar neuron, which is as I described now. We've got the bipolar neuron, where the dendrites and the axons actually look a lot alike, with the cell body in the middle. In the unipolar neuron, where the cell body is actually separate from where the dendrites and axons are connected. You get the Purkinje cell here, very branched out on those dendrites. Makes me think of a Christmas tree. And then the pyramidal cell, which the cell body looks kind of like a pyramid because of how the dendrites are arranged. All right, a simple circuit. So this is where we're going from a perception of a sensation to an actual response. And the most simplest circuit possible involves two neurons. So basically you would have the presynaptic neuron, which is also a receptor and is detecting some sort of stimulus. It then sends the neural impulse down to the postsynaptic neuron, which then receives that signal. This is going to occur usually in the spinal cord or in the brain. And then that signal goes out to signal an effect of a effector, such as a muscle or gland. And it's worth pointing out that the synapse can be an electrical synapse or a chemical synapse. Now usually it's chemical, which means that the axon terminals are releasing neurotransmitters. But occasionally there are places where the axon actually is touching directly the postsynaptic neuron and thereby just passing the neural impulse along electrically. So let's talk about that place where we see this presynaptic neuron contacting the postsynaptic neuron. Basically, there's a lot of interesting structures here. You've got the synaptic end bulb, which is sort of a widening of the axon at its very end. That end bulb is filled with synaptic vesicles that are filled with neurotransmitters, usually of one particular kind. You got the exoplasm inside the synaptic end bulb. You have the synaptic cleft, which is the space between the two cells. And then you have neurotransmitter receptors on the postsynaptic neuron that can then respond to the neurotransmitter and cause an electrical imbalance that will then send a nerve impulse along its cell membrane. So the steps. Step 1, the nerve impulse arrives at the end of the axon. Step 2, this causes calcium channels to open, so the calcium floods into the synaptic end bulb. Then this flooding of calcium ions ends up triggering the release of neurotransmitters. So with the calcium coming in, the synaptic vesicles are now signaled to go to the end of the synaptic end bulb and to fuse with the small membrane, releasing the neurotransmitters into the synaptic cleft. Then step 4, the neurotransmitter diffuse across that cleft and bind to the receptors in the postsynaptic cell. This binding causes the channels associated with those receptors to open, and then sodium usually will flood into the cell, causing a voltage or a charge differential across the cell membrane that then could lead to a nerve impulse that then flows along the cell membrane. So again, this is sort of like a wave. So the this postsynaptic neuron gets a signal from its presynaptic neuron's axon. It causes the change in charge across the cell membrane, and then that change just spreads as ion channels along the cell membrane open up, allowing sodium to flood in, and then closing up when the signal stops. Disorders of the nervous tissue. Well, you got depression, which is where people feel sad, helpless, suicidal. This is linked to an imbalance of the neurotransmitters serotonin, dopamine, and norepinephrine. Major depression is similar to depression, except it lasts for more than 2 weeks. Dysthymia is the episodes of depression that alternate with feeling normal, so that it's sort of a feeling good and then feeling bad. And then bipolar disorder, where you have episodes of depression that alternate with extreme elation. So that's like a major yo-yoing effect. So you feel unbelievably awesome, and then you feel unbelievably horrible. And as you can see, a depressed person's brain actually has depressed brain activity. All right, neuroglia. Neuroglia are other cells in the nervous system. These tend to be smaller than neurons and also more numerous. In the central nervous system, there are 4 kinds: astrocytes, oligodendrocytes, microglia, and ependymal cells. The astrocytes contact neurons, the pia mater, and blood capillaries. So you can see how they're contacting all kinds of stuff. The protoplasmic astrocytes are short, with many branching processes and found in gray matter, while the fibrous astrocytes are longer and unbranched, and they are found in white matter. Their functions include supporting the neurons, forming the blood-brain barrier, helping to maintain appropriate chemical environment, and possibly roles in learning and memory. The oligodendrocytes, they form a protective covering around the central nervous system's axons, which increase the speed of the nerve impulse. Microglia, which are the smallest cells with small little processes, function as phagocytes. They are going to engulf cellular debris, microbes, and so forth. And then the ependymal cells, they form sheets with microvilli and or cilia, and they line the ventricles of the brain and produce, monitor, and assist in the circulation of the cerebrospinal fluid. In the peripheral nervous system, we have Schwann cells and satellite cells. The Schwann cells are flat cells that encircle part of the peripheral nervous system's axons and form myelin sheaths around that axon. And the satellite cells, on the other hand, are flat cells that surround the cell bodies of neurons of the peripheral nervous system. And when you have a bunch of cell bodies together in the peripheral nervous system, that's called a ganglia. Myelination is where the axons are covered in multilayered myelin sheath. This is called being myelinated. It basically helps insulate the axon and speed up the progression of the nerve impulse. Node of Ranvier are gaps in the myelinated sheaths because, well, one cell is not actually overlapping the other, so there's a slight gap between the cells forming that sheath. And the more myelinated axons in the body than there are unmyelinated axons. When they are unmyelinated, though, they're still associated with Schwann cells in the peripheral nervous system. It's just in that case, instead of getting multiple layers, it's basically just holding a bunch of axons near each other. Gray matter and white matter. Well, gray matter and white matter is interesting because they actually look gray and white-ish. The white matter are aggregates of myelinated and unmyelinated axons of many neurons. So you can think of them as sort of the wires of the brain carrying signal. And the gray matter contains the neural cell bodies. And this ends up leading to a sort of grayish color because of the Nissl bodies found in these cell bodies. And this is where the thinking and processing occurs. So you can think of it as the hard drive. Central nervous system has nucleuses, which are a collection of nerve cell bodies, and tracts, which are collections of nerve fibers. Peripheral nervous system has ganglions, collections of nerve cell bodies, and nerves, which are collections of nerve fibers. And you can get multiple sclerosis, which is a disease that causes progressive destruction of myelinated sheaths. It's an autoimmune disorder. It has severe consequences. It's progressive and will eventually lead to someone dying. And that's it for this lecture. Boy, did it take me a lot of speeding up to get done in 15 minutes.